Okay, so this starts our first lecture with this modified flipped classroom. If you remember us talking, when you go on to your Canvas account, so I'm in student mode, it looks a little bit different than yours, obviously, since I'm a teacher, but if you go into your Canvas account under AP Biology, you'll be able to click on modules. Those modules are set up by the units in which we're going to learn. The first day materials are up top, then these active reading guides, which we talked about in class, and then the next thing is your unit evolution, macro, where we're going to start. So the notes for this first lecture are right here, Macroevolution Part 1, Phylogenies, in a PDF version. So if you would like to go ahead, pause the video right now in order to go print off those notes so that you can add to them. You sure can. Otherwise, you can open up your notebook and just take notes by that method, whatever works best for you. But I do encourage you to take notes. This is a lot of information that we'll be talking about throughout the course of AP Biology, and you definitely want to keep things straight, especially terms. So with that being said, do what you need to do right now. You can, of course, rewind this. You can look at it how many ever times you want to torture yourself by listening to my voice. Well, let's get this party started. So, macroevolution. When we think about macroevolution, we're looking at changes that result in new species, which is otherwise known as speciation. So you're going to hear that term throughout AP Biology, whether it's us talking about it in class or even on the AP exams. So those that we take in class, and then obviously the big one that you take in May. So when we look at evolution, we're trying to figure out the evolution history of species, right? Where they all placed together, what commonalities they share, those characteristics, where the common ancestors are, where the derivative points are, and that's called a phylogeny. So if you remember back to sophomore biology, you should have all been introduced to phylogenies, also called cladograms. We're going to talk about the difference between those two terms shortly. But of course, on your screen right here is Mr. Charles Darwin. He is noted as the father of evolution, but there were other scientists that played a role in coming up with what we understand now in terms of evolution. So with this being said, we could spend an entire year on evolution in and of itself, and that goes for every unit, whether it be genetics or if we're talking about ecology. When I was in college, I took one college course on evolution, I took one on ecology, I took one on genetics, I took one on neurobiology. So what we're doing is we're trying to hit the main points, the most important points that have shown up repeatedly on the AP exam, things that you need to know, whether it be background information or stuff that will actually be asked. So we're just going to run through this. First and foremost is taxonomy. Hopefully you remember the concept of taxonomy from sophomore level biology when you did your leaf projects or insect projects. I know you're cringing right now, but you had to list, especially for that leaf project, genus and species. And you all should understand that that is the simplest form of classification that we can get, or the narrowest, I should say, not simplest, but the narrowest. And this classification actually originated not with Charles Darwin, which is oftentimes a misconception with young biologists, but it originated with someone called Linnaeus. Okay, some say Linus, but it's Linnaeus, I say, in the 18th century, so pretty long ago. And he mostly based his classification system on those outward similarities. So he was looking at structural similarities or perhaps inward similarities such as like bone structure, but we're not necessarily talking about DNA evidence or molecular clocks at this point because that was beyond him in the 18th century. So what he did is he came up with a grouping system where the largest category that Linnaeus said was actually the kingdom category. And that was the simplest, the most broad. As you all know, studying biology, we have a broader type of classification, and that's the domain. So you can see domain on the bottom here. Sorry, excuse the little images that point up down there. But so we've got three domains. We've got domain eukarya, we've got domain bacteria, and domain archaea. So with that being said, I could bore you with the fact that he came up with kingdoms within like 1866 for Protista. Monera, also known as bacteria, he derived in 1938. Then he added a fifth kingdom, fungi, in 1969. This was long ago that he was working on this, and from that point on, it's just been refined to better classify all the species that are on our planet. With that being said, you should know also that now when we look at the classification systems, we're looking at those shared characteristics, but on a much deeper level than Linnaeus looked at. So obviously we're comparing molecular DNA sequences and so forth. At one point or another, though, Linnaeus actually knew that he needed to split up the kingdom Monera into two different kingdoms, bacteria and archaea. So basically what you need to understand is that there is this system of classification, which is called taxonomy. You need to know the largest form of classification down to the simplest, the narrowest form, genus and species. And you should also understand or have a decent understanding of those three domains. So when we talk about those three domains, eukarya, archaea, and bacteria, if you're looking at your screen right now, you can see that there is a phylogeny that you're looking at. And interestingly enough, bacteria is the outgroup. Now, long ago, scientists thought that archaea were the oldest form of bacteria. And not that they're not old. Archaic bacteria are often called ancient bacteria, and rightfully so, but actually archaic bacteria share more similarities with eukarya than what was originally noted. So now when we look at a phylogeny between these three domains, we can see that eukarya and archaea are closer on the phylogenetic tree than eukarya and bacteria. If you remember from sophomore biology, we said that archaea, they're usually found in extreme environments. They're oftentimes called extremophils. So we've got those that like to live in very hot temperatures, very salty environments, very low pH. You even have archaic bacteria in the lining of your intestine, and that helps you digest food. So basically when scientists were looking at this, again, to try and refine this taxonomic system, they looked at rRNA or ribosomal RNA to differentiate between the prokaryotes and eukaryotes. Now, if you don't have a concrete understanding between the difference of a prokaryote and eukaryote, you need to pause the video right now and refresh your memory, because that right there is key. That's basic level biology that I'm not going to spend a lot of time on. So if you need that refresher, go take it. So, taxonomic groups from broad to narrow: domain, kingdom, phylum, class, order, family, genus, species. Did King Philip come over for good spaghetti, or whatever kind of little funny saying that you've come up with. And you should know that it's obviously a leveled listing. So when we look at these phylogenetic trees, a phylogeny is said to be the study of evolutionary relationships among a group of organisms. Okay. A tree is usually constructed and said to be like a family tree of sorts. I even use that terminology in sophomore biology. Depending upon the teacher you had, you may have built phylogenetic trees out of pipe cleaners, and we'll kind of revisit that hands-on activity in class. You should know that phylogenies are based on three important things, so the morphology and fossil record. Those, of course, those two studies were not available for Linnaeus at the time. Embryology, so that's the study of how embryos develop over time. So we're looking at similarities within embryonic development among species. DNA, RNA, and protein similarities. So we're really getting down to the very detailed portions of species to try and figure out who's related to who and how closely they are. Now, if you have heard the term cladogram, there's a very subtle difference, and sometimes they're used interchangeably, cladogram and phylogenetic trees. So I'm just going to let you know that a cladogram is basically used to represent a hypothesis, if you will, about the evolutionary history of a group of organisms, whereas the phylogenetic tree represents the true evolutionary history of a group of organisms, the family tree. So when we look at it, probably say both of them throughout class, but the cladogram is the hypothesis of what we think, was the evolutionary history. The phylogeny itself is the true evolutionary history that we've pinpointed thanks to these 3 items that are on your screen. Okay, so with this being said, kind of what I just went over here, we notice when we look at phylogenies that we've got up here, the little dash represents a species or population at any given point. The line itself will represent the population as it progresses or evolves throughout that time. And then the split comes here at a branch point or a divergence point. We call this divergent evolution. This is also called a node, and you'll hear it referred on most multiple choice and FRQ questions as a node. So you need to understand that that's the branching point. So where this species was progressing within a population, at one point or another, it diverged or branched so that now they're 2 different species. Okay, so those traits evolved independently of one another. Okay, but they both have the same common ancestor, which would be found down here. Phylogenetic trees, sometimes what we'll look at is how they were affected by what we call a founder effect, which could be a loss of genetic variation in small populations. So perhaps a species was isolated on an island and there wasn't a large population right off the start, so the interbreeding was a little bit different. We also can look at different selection pressures. So selection pressures can be either positive or negative and influence those populations that way. So we're thinking when we look at selection pressures, what are those? Selection pressures, we're talking about 3 main things. So first, resource availability, their availability of food, water, habitats. So those resources can either positively affect a population or negatively affect it. So that's one selection pressure. Another one that we talk about is environmental conditions. Weather. Were there large events, catastrophic events that started to reshape that ecological niche at the time? A third one is biological factors such as disease. That's a very big thing that threatens populations. We can also look at the impact of mutations as well. So as you know, species can undergo mutations, and those mutations can then affect their young on down the line. And we're going to talk about that more when we get to genetics. Mutations affect germ cells, not somatic cells. So somatic cells are those body cells that we have, like our skin, cells that make up our hair, cells that make up our liver. Your somatic cells are your sex cells. So the only mutations that are going to affect any further offspring, totally blanked, any offspring on down the road has to be a mutation within those, germ cells, the sex cells. All right, enough of that slide. Okay, so if we look at this phylogenic tree, we're looking at a representation of what is thought to be a true evolutionary history of elephants. So you can see where we have, as scientists, where those scientists have posed the Asian African elephant in relation to other mammals and so forth. So the nodes and the lengths of a phylogenetic lineage indicate the time of divergent events. Okay, so this right here would be your outgroup. That is the oldest evolutionary ancestor in this phylogenetic tree. And then as you can see, the length of this line is much different than what we have here. So it didn't take as much time for some divergence to occur throughout this history. And perhaps we could expand this phylogenetic tree to include other organisms as well that could potentially be extinct or classified as extinct. These phylogenetic trees can be oriented both vertically or horizontally. It doesn't matter which. They both mean the same thing. Nodes can be interchanged, so I could take these 2 nodes and perhaps flip them around. I would then also have to place the Asian elephant so that it read and represented the evolutionary history between these 2 organisms properly, but those nodes can be flipped. And we'll look at that when we talk about those pipe cleaners again. On this now, the phylogenetic tree is obviously oriented in a different way. We're looking at other organisms for classification, and this tree in and of itself is going by order, family, genus, and then species. So you can see which ones are more closely related. So by looking at this, we're obviously saying that we're not only looking at those physical traits, but we're also looking at embryologic development, DNA, RNA. Okay, so those are what results in the changing classifications throughout time. So this phylogenetic tree may change depending upon information that's discovered later on. Okay, so let's take a look at this phylogenetic tree, just looking at taxon A, B, C through F. So a taxon is any group of species designated by name. So if we look back here, this here would be a taxon, this would be a taxon, this animal would be a taxon, okay? So taxonomy, keep in mind, is the evolutionary classification going from the broadest domain all the way down to species, okay? And the taxon would be the genus of species. Every node should give rise to two lineages. So if you see here, this branching point gives rise to two, taxon A, and then what takes us to B and C. This node here gives us an evolutionary divergence between taxon B and taxon C. Okay, so that is what is referred to as a true taxon. Okay. Perhaps, though, we have a scenario where a lineage will lead to a divergence of more than two. So this case right here, taxon D, E, and F. As you can see, there's 3 branch points there. That's what's called polytomy. Okay, polytomy is not considered a true taxon. Not that those organisms don't exist, it just perhaps shows us that right now we're still in the cladogram mode where we're giving a hypothesis to how the evolutionary history evolved, and we need to find out a little bit more information about organisms D, E, and F. As highlighted here, taxon B and C are sister taxa. Okay, you've got your ancestral lineage way back here at the left. You have your common ancestor at the first node or the first branching point. Okay, so this is a common ancestor for taxon A all the way through taxon F. Okay, right down here it says that the branches can rotate. We talked about that. So a lot of times what I have seen so far in AP biology tests is that they like to throw in, say, a taxon or a phylogenetic tree like this, but then they like to give you perhaps 3 other choices, and you are supposed to tell me which ones are the same, like they're telling you the same information, and which one is telling you incorrect information. And they flip these branches all over, and they put things there and here and whatnot just to confuse you. But once you understand the phylogenetic trees, it's actually kind of fun. It's like a puzzle. So please note, too, that a phylogenetic tree, not only can it be oriented vertically or horizontally, it can also be in a straight line. And that's typically what we looked at when we drew phylogenies on the board in sophomore biology. All right, so rotating branches. Here's an example. So we've got this that we've been looking at from the start. This here says the same thing. And all we've done is flipped this branching point and this branching point around, top and bottom, but it means the same thing, okay? All right, definition of a clade. So we say a cladogram, that's the overall hypothesis of the family tree, but a clade itself is any taxon that consists of all the evolutionary descendants of a common ancestor. So as you can see by the green, the blue, the yellow, and the red box, each of these different colored boxes represents a true clade. So this green box shows all of these organisms that are descendants of a common ancestor, this first branching point. That is a clade. This blue box shows all of the descendants of this branching point. That is their most recent common ancestor. That's highlighted in blue here. The yellow clade, we get a little bit more specific, just like the red clade. So these two organisms diverged at this point, this node representing the most recent common ancestor. That's another thing that the AP Biology exam will do a lot of times in multiple choice. They ask you for the most recent common ancestor between, let's go back to here, say B and C. Okay, you would not give this ancestor, you would not give this ancestor, you would give this one right here that's highlighted at this node. Okay, so most recent common ancestor versus common ancestor. Be careful in your reading. All right, so when we talk about a true clade, we're looking at those groups that descended from that common ancestor. They're a true group because they're a monophyletic group. They come from one phylogeny. So in this cladogram right here, group 1 highlighted A, B, C. Organisms A, B, C are a monophyletic group because they all come from one ancestor or one common branching point, one node. This is a true clade. Now, in the second picture, when we highlight organisms D, E, and F, they come from the same ancestor here at this node, but they have inevitably left out organism or species G. So we call this a paraphyletic group. So a paraphyletic group is one that has that common ancestor but does not contain all of the descendants. So they left out G. In the third picture, this is called a polyphyletic group. The polyphyletic group highlights organisms that don't have a unique common ancestor. Okay, so as you can see, organism C, its unique common ancestor would be at this node here. Organisms D through G, we could go all the way back to this common ancestor. Okay, that's at this branching point. But in order to basically talk about organisms C through G having a common ancestor, we would have to go back to the oldest, okay, or the first branching point, that first node. All right, so a true clade is this monophyletic group. Okay, that's giving one clade. We've got the paraphyletic where we leave out one of the organisms, oopsies, and then we have the polyphyletic group where we're just grabbing species all over the place. Now, let's talk about anagenesis versus cladogenesis. Okay? Anagenesis is the accumulation of changes in one species that lead to speciation over time. So we are basically going from this cute little birdie here all the way to this bird. Okay? There is no branching that's happening in that realm at all. So there is a progressive evolution occurring between this bird and this. Progressive evolution. Okay, so an evolution of the whole population. For instance, think about giraffes. So at one point, giraffes came from some common ancestor. That common ancestor probably didn't have that long neck. So giraffes, that first giraffe-like species, if you will, evolved over time. It was a progress that occurred to get to the stage in which they're at today, which allows for the greatest rate of survival in order to reproduce. Now, changes that accumulate and perhaps allow that species to become extinct, the first species to become extinct, are part of anagenesis as well. So what they're saying is that this bird here would then be extinct because now we have a better, new, improved, more evolved species there. Cladogenesis is where we have the branching points. Okay, that's also called branching evolution. So you can see that we have evolved from this species here, but it is not extinct. It is still alive, probably. At some point, obviously, species can become extinct, but we're, for all due purposes, saying that this species is still alive, and what happened was this branching evolution where different characteristics became more suited for a certain population. Therefore, we got a different species from the original. So cladogenesis, it says here, is the budding of one or more new species from a species that continues to exist. Okay, so still alive and kicking, just a new birdie came into play, versus this anogenesis where this is now extinct. Okay, so usually cladogenesis involves physical separation. So we say isolation, perhaps. Think of it this way: if a flock of birds were migrating, for instance, and half of that flock stopped on one of these islands on the way, and that flock remained on that island, and then just became suited for that environment. They now will become a different species, okay, with the same common ancestor, but still different species. Whereas the rest of the flock, the other half, they continued to migrate. So now there's some isolation between those two original populations. That results in cladogenesis, okay? All right. Recreating phylogeny. So what do scientists do in order to come up with these crazy diagrams? They look not only at those physical traits that Linnaeus looked at, but they also dig deeper because of the scientific advantages that have occurred throughout time. One of the big things is a fossil record. So what scientists do is they look at that fossil record, and this is just a quick little image that goes along with the understanding. You can read through this image here. What you're going to find at the very end when we look at the sea levels changing, which then results in a different seafloor erosion pattern, where sediments are now exposed, you look and you'll find that the younger fossils, or the most recent animals, are up higher within that sediment versus older fossils, of course, that have been buried over time. Okay? So when we look at, we're going to talk about homologous features and analogous features. So once that group splits into two distinct groups, so right here at this point, so we have anogenesis up until that branching part, and then it becomes cladogenesis. So any feature that's going to be shared by two or more species and then inherited from that common ancestor is said to be homologous. So homologous features are those heritable traits like anatomic structure, DNA sequences, similar proteins, the things that do not change from one species to their offspring. Okay? Hold on, I wanted to mention, I've got my notes here, so you probably hear me turning pages. Let's go back to this slide really quick. One thing that I do want to say in this fossil record is not only do you find the more recent organisms higher up in the level of sediment versus older organisms, scientists use radioactive dating, radioactive carbon-14 dating, and you've probably heard about this before. Basically what they're looking at is carbon levels between carbon-12, which is stable, and carbon-14, which is not stable. So in a living organism, carbon-12 and carbon-14 should essentially equal out each other. So if you think, I mean, we're taking in carbon constantly from the environment, not just humans, but all the organisms on the face of the planet, including plants. So carbon-12 and carbon-14 should equal each other. Once an organism dies, carbon-12 is no longer being replenished. So that level will dissipate and carbon-14 levels will start to increase. So what scientists will do is they'll actually take these fossils in the different sediment levels, like say they grab the fossil from the brown sediment level here in this picture, and they compare it to a fossil that's found within this yellow layer here. They should see that there's greater carbon-14 levels here in this older fossil. Okay, and there's a lot more that goes into it, but basically that's the gist of it all. So just so you are thinking about that as well, that's another technique that goes along with this fossil record. Okay? All right, so basically when it comes down to it, the basis for traditional classification is based on two things: homologous physical structures, and then secondly, the fossil record. So we're looking within the organism, the molecular structure. Okay? So when we talk about ancestral versus derived traits, we're looking at traits that have changed over the course of evolution. Okay? So the original shared trait is called the ancestral trait. And the trait that's found within the most evolved or the newly evolved organism is called a derived trait. So when you look at that cladogram, you can usually, here, sorry for all the flipping, right there. If we take this cladogram, we can look back at, typically we put a little tag here, a little line, a little hash mark, and this would be the ancestral trait. This could be an ancestral trait of this group 1, okay? And then the most newly evolved trait would occur right here that happens before that node where B and C diverged. Okay, going back. Motion 6, sorry. All right, so any feature shared by 2 or more species that is inherited from that common ancestor is said to be homologous. So when you're looking at the image above, human, dog, bird, whale, they have homologous structures because they have similar bones. They have the humerus, they have the radius, they have the ulna, okay, they have the carpals and the metacarpals. Those are homologous structures. Now, sometimes people will get homologous and analogous confused, but homologous structures, what you're thinking of is the same evolutionary origin, okay, but different functions. Now, when you look at this whale, granted, there's the same structures, but obviously the whale is going to use these bones differently for a different function versus a human, okay? We do not swim. Well, we can swim, but not like a whale. You get what I'm saying? All right. So, moving right along, the analogous structures now are those structures that evolved separately from one another. So they have a different evolutionary origin, but basically the same function, okay? So analogous structures, they have similar structure, but they're inherited from different ancestors. So they're not a common ancestor. Just listen to what I first said, because I think I made it worse. And I'm not starting this PowerPoint over again, guys. All right, so we have the humerus, the radius and ulna, finger bones, and thumb. So you can see those highlighted in the bat wing versus a bird wing, okay? So the bones that are found in the wigs of birds and bats are homologous, but the wing itself is analogous, okay? Same function, flight. The wing structure itself did not evolve from the same ancestor, though. So if you locate bird on the phylogenetic tree here and bat, you can see that they have different common ancestors, okay? So, thinking about that, the bottom here where it says analogous structures should not be used in establishing phylogenies. I think at this point you can probably understand why. Homologous structures, sure, we can look at those. Kind of like if you look at the word four, F-O-U-R, like the number 4, I've got 4 apples, versus 4, F-O-R-E, like a forelimb. The spelling is different. They sound similar though, right? 4 and 4, you have no idea what I'm talking about unless I extrapolate on that information. The functions are different, okay? All right. So, analogous structures evolve from similar selection pressures. So earlier on we said that selection pressures were things like resource availability, the environmental conditions, perhaps disease or other type of threatening climates to that population itself, okay? So we can evolve analogous structures in 2 different populations because they're exhibiting or they have exhibited the same selection pressures. Okay, so in this case, we're looking at 2 different animals, the placental mole versus the marsupial mole. So they have analogous structures. Granted, they don't come from the same common ancestor, but their analogous structures are used for the same functions. This is also called convergent evolution. So earlier I had said divergent evolution. Divergent is where when we look at a phylogenetic tree, we see that it diverges or branches off into 2 different species. Okay. Convergent evolution is where organisms are not closely related, so they're not one of those monophyletic groups. So they're not closely related. They independently evolve similar traits as a result of basically having to adapt to similar environments or ecological niches. Okay. All right, another reason why these analogous structures can exist is due to what we call evolutionary reversals. So, for instance, fish gave rise to tetrapods. So, what's a tetrapod? Tetra, you should know, is four, right? Pod or pod means foot, like pseudopod. We talked about that in sophomore level biology, that fake foot, like amoebas have. So, a tetrapod is an organism with four limbs. So the fish gave rise to those tetrapods that were then able to come onto land, such as our amphibians, our reptiles, and so forth. And then our cetaceans, those whales and dolphins, are actually tetrapods that return to the ocean. So an evolutionary reversal. Okay. Just out of curiosity, do you know what we're called? Don't say tetrapod. Okay, do you know? We're called a biped, bi meaning two. Ped is just another version for pod, so walking on 2 feet. So why do analogous structures exist? So we talked about those selection pressures. Take like the dolphin, for instance, and a fish. So the flippers or a fin-like structure was exerted for survival in a certain aquatic environment. So the flipper of the dolphin will be very similar to the fin of the fish. They needed those structures for the same function. Okay, they're analogous. So again, just a quick recap. When we look at homologous structures versus analogous structures, homologous structures have the same evolutionary origin but different functions. So the bat wing, the forelimb of a mouse, the arm of a human. Analogous structures have different evolutionary origin because of selective pressures, but the same kind of function. Bat wing, butterfly wing, bird wing, all for flight. Let's talk about molecular clocks. So molecular clocks, probably we didn't spend a lot of time talking about it unless you had Mr. Coyle for sophomore biology. He loves molecular clocks. So in looking at this image, we are going to be considering homologous structures. So number one, if you look up here, we are showing 2 copies of DNA-based sequences. So DNA 1, DNA 2, we'll call it, of the original gene. Number 2, okay, what do you notice there that's happened? You should notice that these genes, DNA 1, DNA 2, have experienced some type of mutation. So for instance, DNA 1 has experienced a deletion of this amino acid G. What does G stand for? Hopefully you've got that right. If you don't, go refresh your memory on that. DNA gene 2 has gone through a mutation called an insertion, where we took 3 amino acids, which is called a what? Codon, right? And just inserted it in between T and C. So again, if you don't understand what these letters are saying, you need to stop the video now and go refresh your memory. In image 3, we are comparing those 2 genes base by base, showing that no longer do they line up anymore. So up in number 1, they lined up very well. They were identical. In number 3 here, because of those mutations, they're no longer lining up. Now in number 4, scientists will undergo in this molecular clock thinking they will look carefully and analyze via a computer program that will certainly reveal homologous sequences and then start to line them up. Okay, so we can see that there was some type of homologous sequence there. So in looking at this, the molecular clock hypothesis will state that among closely related species, a given gene usually evolves at reasonably constant rate. Okay. So the mutation events are used to predict the evolutionary divergence of species. Okay, if we look at the chart, once we have evaluated the molecular clock, once we look at this chart, sorry guys, a linear graph will represent a constant rate of change, and that's generally what is supposed to happen. Therefore, you can take the slope of that line, and scientists are able to predict how many amino acids would change over a given time span. Okay, So we're looking at hemoglobin here over about 500 million years. So it says the slope of the line represents the average rate of change in the amino acid sequence of the molecular clock. They can then use that and apply it to perhaps changes within another 500 million years. Okay. So what I want you to do is try to predict how many amino acids would change in about 800 million years. Let's say that. So look at the slope of the line. Remember how we determine slope. And see if you can figure out how many amino acids would be changed if 800 million years had passed. Okay? Okay, I'm going to say the answer just so you can see if you're right or not. Okay, so I'm going to say the answer. So if you haven't figured it out yet, maybe stop the video. The answer would be about 1.4. Okay, 1.4 amino acid differences. That's my little guy. Okay, maybe you can't come in. Yes, honey. Hold on. Can you say hi to the kids? Say hi. 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 Okay, go play. They can hear everything we say right now. Hold on. We're almost done. Okay, so this is just a depiction where the divergence points were for that original DNA sequence between DNA 1 and DNA 2. Molecular clocks can also be used to study genomes, like the human genome, for instance. Also, we can look at viruses and how they evolve over time. Molecular clocks have been used by scientists to look at the evolution of the HIV virus. So the HIV virus entered the human population in about 1960 in there. But the origin of the virus was actually dated back to the 1930s, and scientists were able to discover that by use of these molecular clocks. So they do have a lot to contribute to scientific discoveries. If we look at this, okay, this is a lot of reading, so you may want to pause and just kind of examine this chart, because basically this will sum up everything that we talked about and the stuff that I missed with that distraction there. Sorry about that. You'll probably get that a lot throughout the year, or you're going to hear the big German Shepherd barking in the background, so I do apologize, but it's life, right? This is what happens when you make videos at home. So what I want you to focus on right now is the fact that we state here, a stretch of DNA accumulates mutations at a rate that is regular enough to serve as a measure of how long ago two species diverged from a common ancestor. So we can apply that information, like taking the slope of that line from the chart a couple slides back, we can use that and apply it to what we think might happen in the future. We can also use it and apply it to what we're trying to figure out from our past. Over here, what I want you to look at is the DNA from species A and species B. That DNA differs by 4 locations from each other, okay? With that being said, if we now think about species C differs by 8 locations from A or B. So what we're trying to look at when we put together an evolutionary tree is the lineage of species C actually split or diverged from species A and B twice as long ago. Okay, so that is more closely related to the common ancestor than what A and B are. Okay, so we could use this not only to look at perhaps DNA of certain animal species, but we could also look at perhaps strains of viruses, like the flu virus, for instance. You know how quickly that can mutate and change over time. And then the last thing I want to point out here is that if we know the mutation rate, so if we can synthesize that from a molecular clock extrapolation, then we can infer the 50 million year time span from the 4 present day DNA differences. So we can actually infer things from that molecular clock. All right. When we're trying to build a phylogeny, so this is also a frequently asked question when we look at sometimes multiple choice, but more so the FRQs. And you'll see that as we go through, but the FRQs may ask you to actually build a phylogeny based off of a chart of information. Okay, so there's some rules in learning how to construct these phylogenies. One is maximum likelihood and the other is maximum parsimony. So parsimony means coming together, I guess. So what we're looking at is essentially the easiest way to put together this phylogeny to explain the evolutionary history of species in the simplest form. Okay, you want a picture that is easy to read, not a picture that has way too much going on and you lose interest, right? Like I think of, oh gosh, Where's Waldo? I don't have time for that. That's convoluted. You give me like 3 images drawn and I can find out which one is Waldo, okay? The simplest version, right? We don't want to make our lives harder than what we need to. So in this, for instance, we're looking at humans, mushrooms, and tulips, okay? In this first, in A, it's going to depict the percentage differences between sequences of the 3 organisms, okay? So based on those percentage differences between the 3 organisms, we can create 2 different cladograms. Now, the cladogram is going to look at the sum of the percentages from a point of divergence. And what we want to do is you can come up with the 2 scenarios, but you want to pick the one that is simplest. So what they have done is they've looked at tree 1 and tree 2 and compared the possibility of both happening. Well, tree 1 is more likely to happen. It's the most simplistic version between the 2. Okay? So that's what you're going to think of when you go ahead and you build the phylogenies on your own. You're looking for the easiest way to explain an evolutionary history. Okay, this goes in to talk about percentages and whatnot, but what it comes down to again is that tree 1 is most likely to occur. So let's look at this. We are going to build a cladogram from this data table. So this is very, very similar to what you could see on an AP biology exam. So basically you're going to examine the data first. You're going to propose a cladogram depicting the evolutionary history of the vertebrates in the table. What I would do first and foremost, when I'm examining a table like this and I'm getting ready to try and come together with a cladogram or a hypothesis of what I think the evolutionary history is, I want to first see how many organisms I'm dealing with. Well, I've got the lancelet, the lamprey, tuna, salamander, turtle, and leopard. 6 organisms. Great. I know that I need to have 6 organisms placed on my cladogram. In addition to that, when I draw my cladogram, I want to show these different characteristics or the traits that I've diverged over time. How many traits do I have? 5. So now that I've got that figured out, it would be easiest to find out what my outgroup is. So the outgroup is the organism that has the least amount of commonalities with the other organisms. Okay, and what you're seeing here is that the lancelet is the outgroup. Okay, you can see that they don't have a backbone, they don't have hinged jaws, they don't have walking legs, and so forth and so on. So I know that this is going to be the organism that is placed first on my cladogram as the outgroup. And then I know that all these other organisms diverged. Okay, I can see that the lamprey diverged first because now it has 1 trait. It has that vertebral column, okay, versus trying to compare the other ones. And I see that the leopard has all 5 traits. So I know that that is my most evolved organism. Now, not all the time are these charts set up so nicely for you. Okay, this is beautiful. You're not going to see colors. Okay, you're not going to see them in order either. You're going to have to make sense out of all that data. So now what I want you to do is take some time and draw a cladogram for me. I don't care if it's a straight line cladogram or if it's that branching phylogeny that you typically see. Okay, so go ahead, draw that. And then as you can tell in your notes, if you've already got it, here is the answer. Okay, so you're going to place your 6 organisms on there with your 5 diverging traits. So we can see that the vertebral column was the characteristic that allowed the divergence of the lamprey with the other organisms. then we had hinged jaws that evolved, 4 walking limbs, the amniotic egg, and lastly hair. The leopard, of course, has all those traits. Keep in mind, when you look at these cladograms, phylogenies, not only can the nodes switch and read the same way, might not look the same, but it's telling you the same information. You also need to understand that the tuna, for instance, has the traits that are found below it. Okay, so the tuna has a vertebral column and it has a hinge jaw. The tuna does not have 4 walking limbs, amniotic eggs, and hair. Okay, so be sure that you're able to read that cladogram. And we're going to practice a little bit with it in class, but if you don't remember a lot from sophomore biology, I would encourage you again to just refresh your memory on that because it is very, very important. You will inevitably see some kind of phylogeny given to you on the exam somehow, some way, whether it be in this form, whether it be in the chart form, what have you. Not all charts, I just want to point this out, use zeros and ones. Some will use like a minus sign or a dash and then a plus. So we would have like a dash here stating that the lancelet does not have a vertebral column, does not have the hinge jaw, okay? And then when we look at the lamprey, we would see a plus instead of a 1. So the plus meaning the lamprey does have the vertebral column. Then it would be a dash saying it does not have a hinge jaw. It does not have 4 walking legs, okay? All right, so that is lecture 1 complete. All right, so this is going to be how we typically do lectures, just to save us time in class to do those hands-on activities and other labs. The next lecture you will be able to view is the macroevolution part 2. Okay.